Good afternoon. As you heard, my name is Terry O'Regan. I am the president with Brains Bioceutical. And today we're just going to continue with what Dr. Kruber has just been talking about right now. Is there an echo? No? Is there an echo? I feel like I'm in duplicate here. Let me go this side. And we're going to continue the conversation about how to continue to advance the cannabinoid scientific landscape. And to help me do that, I've got a friend and a research partner, Dr. Kishore Alugupalli, who is going to share with you some of the work that they're doing in their cancer research at Therapeutics, and then also just how we can work together to advance the scientific landscape. So I'll be on in a little while, but put your hands together for Dr. Kishore Alugupalli.
So at Screen Therapeutics, our goal is basically to establish Screen 009, a cannabidiol-based formulation, as a part of the therapeutic regimen for standard care of cancer patients. So it's a very big goal. We wanted to actually put the cannabidiol in the front and center in the mainstream therapy. So if you look at here, the US cancer statistics, 2023. So what you see here is, males and females. You see something like a million people get affected every year. These are new cases. And females, again, close to another million. And out of these 2 million, 1/3 are basically going to die. So this is a really very, very serious issue. And if you look at all the 36 major cancers, the top 5 are actually, the melanoma, lung cancer, colon cancer, and of course the gender-specific cancers such as prostate cancer in males and breast cancer in females, and so on. So we really wanted to take these biggies and really kind of subject to our screen-based research. So if you actually look at the US cases and compare to the rest of the planet, what you see is the same story increased by an order of magnitude, 10 times. But if you look at this, basically the landscape of various countries, you can actually see, the US and Europe basically take big part of this pie. And what people are predicting is that by 2040, you see actually 28 million cases as opposed to 18 million right now. So that means this is going to go more and more. So this is not just 1 year. Every year, these millions of cases are repeating almost forever. So this needs to be stopped. So can we do something with this cannabis-based medications? So what is the current therapies for cancer? So you have chemotherapy, which is a cornerstone, which kills good, bad, ugly, right? And then you have radiation, and then you have surgery, and then you have immunotherapy, and you have targeted inhibitors. So you can, if you understand the specific pathway, you can go with these small molecules that can basically break the pathway. And what is the major problem with the chemotherapy? For example, if you take chemotherapy, 75% of the people with chemotherapy are going to suffer a pain called chemotherapy-induced peripheral neuropathy or chemotherapy-induced neuropathic pain. So this actually happens in the first 1 month. You can see 75% of the people, and regardless of the chemotherapy agents, and which actually goes down by 3 months, it can go down by 50 to 40%. So the bottom line is that this is a very important side effect one has to deal with it. And so what is the problem with cannabidiol here is that while this has an amazing molecule, has an extraordinary potential, it has a few limitations. As you all know, the poor water solubility, low bioavailability, suboptimal bioactivity, so that you can actually develop a nice formulations like we have developed and many people have developed. So I wanted to acknowledge the field in general. And there is a very different pharmacokinetics and pharmacodynamics in between males and females. So these are all important. So what did we do? We basically made a formulation, which is basically a nano formulation of 100 to 200 nanometer nanosomes, and you can actually use this thing. These are water soluble, you can adjust the dose, you can give oral, and even these are can be given injectables. And what you see in this case is that I wanted to kind of show you the proof of principle with this formulation. We actually taken the most biologically relevant melanoma model, which is basically BRAF mutation. So 50% of the melanoma has a BRAF mutation in the 600 amino acid position of the BRAF, the valine to glutamic acid. So single mutation. And you actually can take these mouse melanoma and actually implant it as syngeneic mice, means the same genetic makeup, and then you can see actually how the tumor grows. And what you see in this red line is that without any treatment, you have a very nice growth, as you can see on the x-axis versus the y-axis, the tumor growth. This is how a progression happens. If you were to treat these animals with CBD without any formulation that we have done, you can actually delay this progression, as you could see in the open circles. But if you make this formulation, is actually much more efficient. So the idea of the bioavailability of the potential bioactivity has been significantly increased while keeping the CBD concentration exactly at the same. So one of our colleague and collaborator in Philadelphia, Dr. Sarah Jane Watt, actually did a wonderful study in the CIPIN models, in the rat models as well as the mouse models, where she has actually shown that you can actually measure the pain by measuring the mechanical threshold of whatever the one you're using to kind of cause the pain. So if you see on the very first panel A, you can give the IP, the CBD, obviously these are treated with oxaliplatin, platinum-based chemotherapy. And as you could see, when you treat with more and more concentrated CBD, you can actually bring this mechanical threshold back to normal, indicating that you can actually decrease the pain. And you also can do the same thing in the oxaliplatin-induced prevention. So in this case, you basically treat them before, so you can actually prevent them. And she also has done with paclitaxel and paclitaxel prevention in both models. So what you do in this case is that whether you inject IV or oral, you can actually prevent chemotherapy-induced neuropathic pain very nicely. So having shown the first part of this particular formulation, we actually hypothesized that the formulated CBD is going to actually work much more efficiently for the chemotherapy-induced neuropathic pain. But now there is a problem. So what happens if you really wanted to do the CBD treatment in the chemotherapy context? So is it going to be compatible or not? That is a fundamentally very important question. That's precisely what we have addressed. We actually took the melanoma, the BRAF melanoma, and what you see in this case is that no treatment, as you could see, the black dots, kind of going up with the faster kinetics. And if you give the dacarbazine, one of the choice of chemotherapy, you can actually decrease them. But what surprised us is that the 2% CBD containing serine formulation or 6% worked actually outcompeted significantly in statistics, you can actually see, than the actual chemotherapeutic agent. This was surprising. But when we actually put the dacarbazine and the serine 009, what you see is a really kind of inhibited the tumor progression significantly. So this is extraordinary. So in basic research, because I'm actually a member of the Kimmel Cancer Center, one of the NCI designated cancer center at Thomas Jefferson University. So one of the way you can actually read out is that look how the growth, the tumor growth is happening, and then certain growth, when it happens, you have to euthanize the mice because there's no point of return. So that you can use it as a survival. So that is how all the drugs are actually been approved. So what you basically do is that you actually monitor the survival curve. So you can see the black line in the panel B is that by 35 days or 33 days, all the mice basically die. But when you treat with just the CBD containing serine 009, you can actually increase the survival by 7 to 8 days. This is really an extraordinarily improved survival. And then what we wanted to do is that can we do for any other cancer, and it is just for the melanoma. So what we basically did is that we picked yet another cancer, so the lung cancer model. This is what actually the model that made the approval of a lot of lung cancer drugs, including the chemotherapy. And in this case, the mutation is a KRAS mutation, classic KRAS mutation right downstream of the EGFR receptor signaling. And even in this model, what we actually saw is that you can see the black one, without any treatment, kind of tumor goes on. And when you have the carboplatin, in this case, a platinum-based chemotherapy is actually the one that is choice. And you can see the blue one is actually kind of working pretty nicely. But again, even in this model, SRIN-009 actually outcompeted the efficacy of the chemotherapy agent. But when we put both of them, we got an actually additive effect. So by showing these two models, what I'm trying to kind of convince you is that the cannabidiol-based medication is actually compatible with the chemotherapy. And also one more important thing you notice here is that if really the SRIN-009 is this efficacious, you can probably decrease the actually chemotherapy doses. By doing so, you can actually eliminate, eliminate a lot of chemotherapy-induced issues, or at least you can minimize. This is something very, very important point. Again, we did the survival curves. You can actually again see, you have a significantly improved survival even in the KRAS lung cancer model. We went on to go for yet another aggressive cancer model, that is a colon cancer model. Colon cancer is a very aggressive model. And even in this aggressive model, we have actually shown that SRIN continues to kind of decrease the progression of the tumor, and the survival is also increased. So this is great. So certainly what I'm trying to kind of show you or convince you is that a cannabidiol-based medication is completely compatible with the chemotherapy. And then we wanted to kind of look what kind of effects the CBD-based formulation has. As you know, the CB2 receptor is on the immune system. By training for 30 years, I'm an immunologist. So we wanted to really kind of look for the immune-related functions, because immunotherapy is actually becoming mainstream for a lot of cancer treatments. So what we basically did is that we have treated the mice with various chemotherapy agents as well as SRIN-009. What you see here is that each dot represents one animal, and you can see the erythrocyte count is something like a 10 on this particular scale, and you can see the chemotherapy agents depending on what it is. You have a significantly decreased red cell count, suggesting that the erythropoiesis is significantly inhibited. As you know, chemotherapeutic agents work very efficiently on a cycling cell. So as you know, the erythropoiesis is being affected. Unlike the chemotherapy agents, the SRIN-009 at 2 different concentrations has no effect. We also measured the hematocrit, which did not change. And we also went on to look for the lymphopoiesis, both B lymphopoiesis and T lymphopoiesis, because these are the ones actually play a very important role in tumor control and tumor surveillance. And even in this case, what you see is that the lymphocyte count was altered by the chemotherapeutic agents, but not by cannabidiol-containing SRIN-009. And now what happened, what I showed you is that the cell count significantly went down, but all these particular mice that are treated with SRIN are actually immunocompetent. So what we basically did is an immune response to a foreign antigen. And in this case, we have used a foreign antigen. And this antigen response is driven both by the B cells as well as the T cells, and some completely germinal center-driven response. In this particular case, as you expect, doxorubicin, as you can see, lymphocyte counts went significantly down, so quantitatively the immune response should go down. So that's what probably you see here. But SRIN has no effect, showing that it is compatible with the immunotherapy. So that is the third thing I have actually shown. So here is basically pretty much my summary, is that the SRIN is compatible with chemotherapy. I showed you we wanted to basically go after the radiotherapy, and we also work on the chronic surgical pain. So we wanted to kind of see whether it's also compatible with the surgery. But those surgeries are not a painful surgeries, otherwise, unlike other surgeries. I showed you the immunotherapy compatibility, at least expected immunotherapy compatibility, but it will also be compatible with the targeted inhibitor because I have shown you 3 different models where the mutations are totally different, yet the efficacy is remarkably fantastic. So our ongoing actually clinical studies are going with various indications. CPIN is actually one of the driver, and we are actually ready for the phase 1, phase 2 clinical trials. For this particular thing, obviously we need the partnerships, and today I had a wonderful interactions with Dr. Deborah Kimlas, and we wanted to really work. She is also a Philadelphia person like me, and we are going to collaborate. And one more important thing I wanted to do is that I wanted to acknowledge Alugupalli. He is the one actually founded this company in 2018 and recruited me a week after. And the second is me, and we have a CMO, Dr. Vijay Sandilya. He is the CMO, and he's our in-house hematologist and oncologist. And our collaborator, Dr. Sarah Jane Ward, and another colleague of mine at Jefferson, Brooke Worcester, some of you probably know, and she is actually the director of the cannabis medicine at Jefferson, and she's really kind of pushing this cannabis medicine amazingly. And she's also the new director for the supportive medicine and cancer survivorship at the Thomas Jefferson University Hospitals. And last but not least, I wanted to kind of thank Terry O'Regan and Brains Bioceuticals, because they are the ones going to provide us the API grade cannabidiol for the clinical studies we are going to go forward. And with this, I wanted to kind of ask you if you wanted to collaborate with us. You are more than welcome, and we are also interested in any venture partners that wanted to join us.
Thanks very much, Kishore. All right, everybody, and so I'm just going to conclude this very quickly, and I just want to talk about how we can collaborate together to advance the cannabinoid scientific landscape. And why that is so important to me is because I come from the pharmaceutical background. I have been in pharmaceuticals for all of my career, and I've joined Brains 18 months ago because so many oncologists and neurologists and psychiatrists said, Terry, we know it works, but we just need somebody to do the clinical studies to tell us how it works, where it works, and how to use it. And that's what we're trying to do here at Brains Bioceuticals. So, I'll go straight on. This is the challenge. I am so grateful for everybody that is providing medical cannabis at the moment, but I come from London, and it's the 2nd biggest medical cannabis market in Europe. And when I look at the statistics, only 1% of patients are getting access to medical cannabis from those that need it. All right, so it isn't the solution. It is a solution. It is making progress, but it's not the solution. And that is where we go to, I come from a pharmaceutical background, and where we're saying, okay, let's play by the rules that pharma understand, the FDA understand, and let's try and advance this in a different aspect to what some of you are doing, but together we'll get there. Ultimately, it is about giving patients access to these treatments. So what we do is with our collaboration partners, Lakshman, also with DSM and Fumanish, is we look at all the aspects of a good clinical program. And, it sometimes breaks my heart to see these failed studies because people did some guesswork or they didn't do enough due diligence. And so what we're looking at is how we can provide this end-to-end service, in terms of the input material, start right from the start, and then going into making sure that it is a commercially viable asset that you can progress and get funding for. Our partners, DSM, are world leaders when it comes to formulation. So how can you formulate it in a way that is conducive to the study that you're doing, as well as potentially strengthening your patent? And then how can you reduce the cost of clinical programs, looking at decentralized ways of doing clinical programs? And, we're in the business of isolate because that's what the FDA understand. They want to know exactly what is being combined with what. And so we are making it, we're doing an API, starting with our CBD, but soon to have other cannabinoids, where we're looking at how we can potentially provide the best API available so that we do not compromise the clinical studies that you guys are involved in. As you can see, we can guarantee consistency as well as being RCHQ7 compliant, as well as German DAC pharmacopoeia compliant. So when you want to get into Europe, we meet those standards. And then just coming into a close, we've got a reference DMF, so for people doing clinical studies. We've also got an ASMF in Europe, and we're looking at bringing other cannabinoids to the market. My final slide is from DSM. As I said, they're world leaders in formulation, and I know that is kind of a big challenge, but they're looking at ways in which we can improve the bioavailability, improve the stability, improve the loading, and they are pioneering in this area. And Alex, my colleague, has a poster outside if you want to know more about that. But finally, I think, I heard Brendan's story last night about his father, and I heard Bruce's story today about his wife who had ovarian cancer. And as I go through, I hear all the stories as to why we got into this. And all I can say is it's an honor for me to step into the sector to work with you. And we're all doing our part. Some of it is different, but we're all doing our part because ultimately, leaders leave people in a better place than when they found them. And I know together, you're leaving patients in a better place than when we found them. Thank you for your time.